So you're going to work on a new job. Final wax at some point. Okay, good morning. Why don't we get started? So we've been talking about RTKs, receptor tyrosine kinases, and sort of our poster child for this family has been epidermal growth factor receptor. But I did want to mention one of the really important clinical examples of this family, and that's the insulin receptor. It's important not only for diabetes, but it's also important for cancer, because a lot of tumors overexpress insulin receptors because, as you'll see, it's quite a growth factor as well as something that stimulates glucose uptake. So the question that we're going to try to answer is this major learning goal here, which is how does the insulin receptor do both of these things? How can it simultaneously increase cell number and also increase glucose transport, which is, you're familiar with that aspect of it. But what we haven't shown is how does it get from insulin binding to an increase in GLUT4 disposition onto the plasma membrane. So that's really the goal of this part right there. And it's probably the more complicated of the two features. Okay, so we know this part, and this is the MAPK pathway that we talked about last time. And so the insulin receptor can serve as a scaffolding and an activator of that MAPK pathway. Okay, so the only thing that's wrong here is the fact that this is shown as a monomer, and as you know, it has to be a dimer. And so the insulin receptor is kind of interesting because it is constitutively dimerized. It has an alpha and a beta subunit, okay? And actually the role of it, so it's always dimerized, so you would think, well, it's always on then, right? No, the reason it's not always on is this alpha subunit is actually an allosteric inhibitor of the beta subunit. The beta subunit is the one that spans the membrane like any classical RTK with a single transmembrane alpha helix. Okay, so this is the beta subunit. That's the tyrosine kinase, and it is turned off as long as the alpha subunit is unoccupied. Okay, so to relieve the inhibition of the alpha subunit, what has to happen is one insulin molecule has to bind. Okay, and what that does is it changes the shape of that alpha subunit, relieves the allosteric inhibition of the beta subunit. This turns on, and what does it do? It's going to cross-phosphorylate like any other reasonable RTK. All right. So when you do that, what's going to happen is it's going to coat itself with phosphorylated tyrosine residues, and that serves as a binding site, as you now know, for Legos to stick on them. Those Legos have to have what kind of domain? Here's some rumblings. What's the Lego that sticks to phosphorylated tyrosines, phosphotyrosines? SH2, right. Sarc homology 2 domain plugs in, okay? And so what actually plugs into the insulin receptor is this large molecule. It's called, this is actually called, it's not the Internal Revenue Service, it's the insulin receptor binding substrate, insulin receptor substrate, and it's got different isoforms, which we're not going to worry about. So IRS1 is insulin receptor substrate. Why do we need this other docking protein? The reason we need this thing is there's going to be so many pathways that branch out of the insulin receptor that we need more physical space. So what happens is when the IRS1 binds to it, it's actually holding on with two different hands. First of all, this is actually not an SH2 domain. It has the same function in that it binds to phosphotyrosines. But technically it's different. It's recognized a slightly different set of amino acids on either side of that phosphotyrosine. Is it in the same family as an SH2 domain? Absolutely. But we call it a phosphotyrosine binding domain, a PTB. But that's just the same thing that an SH2 does, is it's a phosphotyrosine binding domain, right? But this is a little bit different from an SH2. It does the same thing. It plugs it in to the insulin receptor if the insulin receptor has been activated by what? By insulin. Okay. All right, so now we have this big scaffolding. The other hand, it's sort of like a monkey hanging onto monkey bars in one hand and onto a tree with the other. The hand that's hanging onto the cell membrane is interesting. It's another Lego, but this Lego has a different name. It says pH and has nothing to do with acidity. It's a plextrin homology domain. And I've got it, it's over here. It was actually misspelled in another, so let me point that out. So pH domain is a, where is it? Okay, well, I'll find it here in a moment. But it's a plextrin homology domain. There it is. And it's still spelled in this one. Right there. So you should put a little S right there between the K and the trin. So plextrin homology. So what is that Lego looking for? It's got to be looking for a binding site. And it's looking for a phosphoinositol phospholipid that's phosphorylated in the 3 position. That's what it's looking for. It doesn't matter what the other 2 phosphates are or if it's just got 1 phosphate, but it has to be that inositol lipid head has got to be phosphorylated, let's go, in that position. That's the 3 position. Okay. So you're going to have some kind of enzyme that actually phosphorylates these phosphatidylinositols in that position to recruit the insulin receptor's activity. All right? And one of the first things it recruits is IRS1 to give itself some binding area. Okay? So that's what it's looking for. It doesn't really care about these other ones over here. It's looking for that phosphate in the 3 position of what? The cytoplasmic leaflet of what molecule? Phosphatidylinositol with that little head group. Okay? All right, so that's one way that IRS is holding on to it. It brings IRS to the membrane. It attaches to the receptor. And then it's going to recruit the very standard MAPK pathway, okay? And so you know this story, right? So you have GREB2, instead of binding to the receptor, which it could do, right? But it actually has more room to bind to this thing, because what the insulin receptor does is once it's recruited the IRS to it, it just peppers it with all kinds of phosphotyrosines. This thing is coated with all kinds of tyrosines that can be phosphorylated. And so the insulin receptor is the one that puts all of these phosphates onto IRS1. Okay? So that provides all these docking sites. And so the first thing it binds is GRIP2. You know that story, okay? And then it has 2 domains. It has SH2. It has an SH3, which is a polyproline binding site. And so our old friend, SOS, Sauce, son of Sevenless, has a polyproline tail, and so it will bind to SH3. And in mammalian cells, unlike Drosophila, it actually has another hand group, just like IRS1. It has a plextrin homology domain, and that's really convenient because it's going to put it right next to the plasma membrane. Why is that a good idea for SOS to be there? Why would you want SOS to be at the membrane if it's turned on? What's its binding partner? What's the next one in the series? RAS. And RAS, as you know, is an old friend of ours, has a lipid anchor. And so RAS must be in the plasma membrane. So if you recruit sauce to the membrane, that's the GEF for RAS. That's the RAS GEF. And so you're going to turn on RAS, which is going to turn on RAF. You should know these. RAF, MAP. And MAP does what? It goes into the nucleus and stimulates transcription to send the cell into the cell cycle. We're going to talk about that in the last lecture a week from today. Right? All right, so we're going to turn the cell division on. So this is how you activate transcription. Okay. Now, we haven't said anything about glucose transport. And that's because that spins off a different pathway. It's going to actually come off of IRS1, okay? And the way we start that pathway is we recruit this guy. This is phosphatidylinositol 3 kinase. You might remember that this is one of the RAB effectors that help RAB do its job, okay, in phosphorylating specific sites in the donor and target membrane. Well, here, what it's doing is it's creating lots of binding sites for proteins that have pH domains, flexible belongs. Okay. So it's going to give IRS1 a place to hold. Its target is to create this phosphatidylinositol with that 3-position phosphorylated so that it can recruit 2 key proteins. The first one is called AKT, and AKT is a serine threonine kinase. Okay. Remember, if you see a kinase, you need to know what it phosphorylates. So this is a serine threonine kinase. This is a serine threonine kinase. It's called PDK1, and I've defined what that stands for. It's a mouthful. We'll just leave it out. But both of these have plexural homology domains. Okay? And the sole reason that you've been phosphorylating the cytoplasmic domain is to recruit both of those to the cell membrane. Okay? So now, this is at the cell membrane. AKT is a coincidence gate. Okay? Remember we talked about a coincidence gate before, which would be PKC. Remember it had to have DAG in one hand, and it had to have calcium in the other. It required 2 things, right? This one requires PDK1, and it requires a second kinase that's cytoplasmic called mTOR. Okay, that's we described it over here. Okay, so it requires both of those to be active. This is a really dangerous pathway because it causes cell proliferation. Okay? Lots of tumors love this pathway, and what they do is they activate this pathway sometimes without any mTOR or without any PDK1 because they've undergone activating mutations such that AKT is on all the time. That's really dangerous. Okay. So this is highly controlled because it's exquisitely important for cell growth and metabolism. Okay, so we've got to have 2 prongs that are coincident in activating one kinase, and this is a serine kinase. So what's the big deal? AKT does a lot of stuff. Once it gets activated, it releases from the cell membrane, and let's look at this pathway first, okay? It's actually going to activate the movement of GLUT4 from GLUT4 storage vesicles to the plasma membrane. So now you're getting the rest of the story. How does it do that? Well, so your book goes, okay, here we go. Here's the GLUT4 storage vesicles. So here they are waiting. So this is part of the regulated secretory pathway. What's it waiting for? It's waiting for insulin, right? Because let's say this is skeletal muscle, okay? Skeletal muscle is the major consumer of glucose after you eat a meal, okay? So if you had some sort of, whatever, bagel or something this morning, where is that glucose going? It's going right into your skeletal muscle if you have adequate insulin secretion. So insulin's going to bind, and it's going to go through that pathway we just saw. Activate receptor, activate recruit, PK3 kinase, phosphorylate the membrane, recruit PDK1, recruit mTOR, recruit AKT, turn on AKT, phosphorylate this. And that sends it to the membrane, and you get a 40-fold increase in the delivery of GLUT4 to the plasma membrane. And glucose pours into your skeletal muscle, and where does it go? Goes into your fat life. Well, if you're just sitting around, what's going to happen? That glucose is going into your skeletal muscle, what's going to happen? You just pee it out. Pardon me? To. What's the storage form of glucose? Thank you. You store glycogen granules. So when you get up and run across the room, you can burn that stuff. That's the first readily available carb that goes. You burn carbs first, then you burn fat, and if you're about to die, you finally burn protein if you've been in a concentration camp for 30 months. Okay, but the first thing you reload and unload is glucose, glycogen, glucose, glycogen. Okay. All right, so this is key to all of it. Right. Okay, let's step back and look at this thing. So remember, there's actually two ways that insulin causes cell numbers to increase and why it's so dangerous in terms of being a cancer promoter. So the insulin receptor is a oncogene tumor suppressor, which is, there's two choices. Is it an oncogene or is it a tumor suppressor? Okay, I would think about that before the test. Okay, so here we go. We've activated the MAPK. That's going to drive cells into the cell cycle, but there's another way to get cell numbers to increase. Stop apoptosis, that is programmed cell death. So if you block apoptosis, you can get cell numbers to increase. Because you're increasing the number of cells, but if the number of cells that are killing themselves equal each other, you're not increasing cell number, right? So if you're increasing cell number and you're suppressing cell suicide, your cell numbers are going to go through the roof. Okay? So that's why this is a very dangerous pathway. Because what AKT does is it phosphorylates a protein called BAD. Okay? And what happens is BAD is holding on to, when BAD is doing its bad boy thing, or bad girl, I don't know, whatever it is, it's holding on to an apoptosis inhibitory protein. Okay? And what it's doing is it's preventing apoptosis from being inhibited. So it's allowing cells to kill themselves. That's why it's called bad, because it stimulates apoptosis. But apoptosis can be bad in itself, right? Okay, so, but apoptosis, control over apoptosis controls cell number too. So what we want to be able to do is control it. And what happens is when AKT phosphorylates BAD is actually tied up with another protein called, strangely enough, 1433. Oh, that's a wonderful name. Okay, so what it does is it binds to phosphoserines and activates BAD, and look what it releases, this little torpedo here. Okay? This is an active apoptosis inhibitory protein. So this is now going to block apoptosis. And if you're a tumor cell, you would love this to happen, right? Because now cells are not only being stimulated by the MAPK pathway to increase, you're preventing your cell death. Right? And so that is very dangerous. Right? And so you can see now, why would tumor cells overexpress insulin receptors? Number one, because they activate the MAPK pathway. Let's go back. Turns this guy on, and you know the pathway is SOS, RAS, RAF, NEC, FAT. Goes through the nucleus, cell cycle goes over and over and over, you get more cells. And then meanwhile, you stimulate this pathway to activate AKT. And of course, you know, in skeletal muscle, it doesn't undergo a lot of cell division. But if this is in a tumor, what it's going to do is phosphorylate BAD. BAD gets tied up. It can no longer prevent, it can't prevent this inhibitor of apoptosis from working, so it releases this little torpedo, which is an inhibitor of cell apoptosis. So it's one of those double negative things. So if you inhibit the inhibitor of apoptosis, what happens is this, if you release this guy, it's going to prevent apoptosis from happening. Cell number increases because you're not killing them off. Okay. So let's go back. I mentioned to you that one of the cancers that I was studying, which is cutaneous T-cell lymphoma, if you give them HDAC inhibitors, that suppresses the cancer completely, you know, turns that patient around. 7-8 months later, it comes right back. Okay? And so the reason is they become resistant. And they become resistant to the HDAC inhibitors. And why is that? Well, it turns out we discovered that reason. So you can think of a lot of reasons why that tumor would have been resistant, but what we discovered first was the insulin receptor was overexpressed. So you go, aha, it must be that the MAPK pathway is stimulating more cells, cell division, and this, we really suspected this first. The AKT was overexpressed, and it was allowing apoptosis, it was blocking apoptosis by phosphorylating bad and releasing this inhibitor of apoptosis. And so the apoptosis was going wild, and the patient's tumor was just going nuts. Right? Okay. It turned out when we assayed for that, you don't assay for AKT, you assay for phospho-AKT. Right? That's the active form. You have to be sure to look for the active form. It wasn't increased at all. Nothing. Okay. So the insulin receptor's up. So we go, oh, well, it must be MAP must be overexpressed. And it was up a little bit high, but it turned out what actually was happening is MEC was way overexpressed. It's the step above MAP. And what MEC does, it does the same thing as BAD, or it does the same thing as AKT. It inhibits an isoform of BAD called BIM. And so there's a dizzying array of these isoforms, but it was doing that. It was inhibiting BIM. It was tying up BIM just like this, and BIM was releasing its version of this apoptotic inhibitor and blocking apoptosis in those patients who died, actually. So the treatment was to give both HDACs and MEK inhibitors simultaneously, completely wipe it out. Cancer's gone. Okay. So this type of pathway is useful in both cancer studies and also in diabetes. What's the deal with diabetes? Okay, well, let's talk a little bit about type 2 real briefly since we're looking at this. One of the problems, let's say skeletal muscle, is in type 2 diabetics, they become insulin resistant. What does that mean? It means the insulin signaling pathway is not working properly. Okay? And you know that this is sort of associated with obesity. So what's the connection between obesity and diabetes? Well, it turns out that if you have a certain type of obesity where you have lots of fatty acids in your bloodstream, they get stored in the wrong place. Where are you supposed to store fats? What tissue? Not your brain. Adipose tissue. That's a healthy place to store excess calories. But what happens is in type 2 diabetics, they store it in skeletal muscle, in the beta cell that produces insulin, and in the liver. Those are terrible places to store excess fatty acids. So let's talk about it. If you store it in, so let's pretend this is skeletal muscle. What's going to happen is the fatty acids go in, and they're stored there, and those fatty acids create excess DAG. Okay? So the DAG levels in the membrane go up. That activates PKC, a really specific isoform called PKC theta. There's a bunch of these different isoforms. And what it does is it activates PKC. What kind of kinase is PKC? Tyrosine, serine threonine, dual specificity? Which kind is it? You got three choices, take a guess. Okay, click in. Let's make this a clicker question. A is serine threonine. We must be asleep this morning. Okay. Let's say A is serine threonine, B is tyrosine, and C is both, dual specificity. This is an attendance question, so don't worry about getting it right. But click in something, okay? Is it open? It's not open. All right. Actually, just do the first two because I think I only have two answers there. So it can either be serine threonine or it can be tyrosine factors. That's what I wanted to do. With the clock. Oops. 16 seconds. Okay. Ooh, all right, so the answer is A. Okay, it's serine threonine. Okay. And so what that's going to do is it allows it to phosphorylate something that's not going to help out. And the thing that it phosphorylates is that. PKC in diabetic skeletal muscle will phosphorylate the insulin receptor. And serine or threonine phosphorylation is a off switch for the RTKs. Okay? And so it desensitizes the insulin receptor, so it takes more insulin to get the signal to them, because this is coated with all kinds of phosphoserines, which recruit proteins that turn it off instead of turning it on. So the on switch is phosphotyrosines, the off switch is phosphothreonines and serine. And so that's the relationship between obesity and type 2 diabetes. And we could talk endlessly about this, but I continue this conversation in endocrinology next semester. Okay. All right, any questions about this fairly complicated story? Okay, and this is just to show you what's actually happening is some of these proteins get phosphorylated, and in some cases we're sure what they are, others we're not, but it removes these inhibitory proteins that allows the T snares and B snares to form a 4-helix bundle, and then you get delivery of GLUT4 to the plasma membrane. But this phosphorylation step is absolutely required. Okay. All right, so the take-home from all this signaling stuff is it's really complicated, and there is actually, several things branching off the RTK. Meanwhile, in the same cell, you can have a number of G-protein coupled receptors also talking to the cell. And so the ultimate cell response is the combined response to all of these signals and their crosstalk. So that's what makes this a very complicated story. Okay. All right, so that finishes cell signaling, or we have to leave it at that. And this background, though, should allow you to dissect any kind of cell signaling pathway that you're faced with. You look for the receptor type, you figure out the pathway, and then you know the signaling pathway. Okay. All right, so let's move on to our next to last topic, which is cytoskeleton. Fortunately, the cytoskeleton is a very, very dynamic topic. And we need another PowerPoint. Let's see. Yeah. Okay. So, first thing, we're going to have to cover quite a bit of stuff fairly quickly, and so what are the major things that you need to know for the final exam? Well, the first thing is to be able to compare the 3 major fiber types in the cytoskeleton and answer a few simple questions about how they differ and how they're similar. So that's what we want to know, is how they differ in structure, assembly, function, and stability. Those are the big questions. And then we're going to get into some of the specifics of the different fiber types. Okay. So this is a nice, nicely stained cell because it shows all 3 different fiber types. The greens are microtubules. Okay. And you notice they almost always radiate away from the nucleus, and there's a reason for that. So they tend to go from the nucleus towards the plasma membrane. These yellow fibers are called intermediate filaments because they're intermediate between the largest fiber type, which is microtubules. Even though the name is micro, it's actually the largest fiber type in diameter. Intermediate filaments have a diameter that's slightly smaller. okay? And they actually parallel the microtubules pretty much, not exactly, as you'll see. And the ones in red, these are the microfilaments, and they're the smallest diameter fiber. So one way we can tell them apart is by the diameter of the fiber itself. Okay, so there's a very structural relation. And the cell distribution is very different between microtubules and microfilaments. Microfilaments tend to be highly concentrated right underneath the cell membrane. That's called the cell cortex. And that's where you find most of the microfilaments. Okay. So let's go through some of the basics, right? So here's a cell, and its cell shape is strongly determined by two things. The cytoskeleton is what determines its shape, and which filaments do that? Mainly the actin microfilaments. So actin makes up microfilaments, and that helps determine whether the shape of the cell is spiky, or it's got a pseudopod, or whether it's round, or whether it's moving in some direction. Okay, so it determines the cell shape. The microtubules also play a role in determining cell shape because they're actually little superhighways by which all the cell components are being trafficked. You know, the Golgi apparatus is sitting right on top of microtubules moving around. Okay? Vesicles are hitching a ride on this microtubule superhighway. So in a very real sense, all these components have to follow this little green highway, which are microtubules. And how can you tell that's the microtubule? Because it always originates near the nucleus and then radiates outwards. Okay, so here's a dividing cell, and most of you know microtubules make up the spindle fibers. So clearly the role, a really clear role in chromosomal alignment and segregation is the spindle fibers, and they're made of microtubules. Whereas, so that's mitosis. What's this process right here? It has a specific name. When you pinch off, what do you say? Cytokinesis, right? That's not mitosis. It's cytokinesis. Pinching off is driven by a little belt of contractile tissue, just like skeletal muscle. It's made of actin and myosin. So this is microfilaments that are causing that little belt to squeeze and pinch off the two cells. Okay? And basically, so what about the intermediate filaments? Intermediate filaments provide the basic structural stability in a cell. They're there to keep the cell from being pulled apart. So really, intermediate filaments are there for structural integrity and not much functional. Okay. Okay, so here's a picture that sort of helps illustrate this. This is an old friend of ours. These are enterocytes in the gut, right? And here's Bart Simpson's hair. So this must be the gut lumen. This must be the blood. Okay, and so the reason you have Bart Simpson hair is because microfilaments are poking up through the cell membrane. That creates this increase in surface area. And why do you need that increase in surface area? Functionally. I'm sorry? Well, and then so that would be true for simple diffusion, but what are most nutrients absorbed by simple diffusion? No, they're absorbed by transporters. You need all that surface area to pack all the proteins into the plasma membrane. It is standing room only up in there, okay? And what gives it that extra surface area are protrusions of microfilaments, okay? So again, determine cell shape, and look at, now this is a really odd example of the way to arrange microtubules. This is not typical, okay? Because they don't originate from the nucleus, they originate actually from basal bodies that are underneath the cell membrane, and they run in a very polarized fashion. Notice that they have little pluses and minuses. Those are not electrical charges. Those are the polarity of the filament itself. So some of the filaments, most of them, microfilaments and microtubules, have a structural polarity. There's a minus in where they start, and then there's a plus in where they leave. Okay? And so we'll talk about the significance of that plus and minus, but it has nothing to do with electricity. But this is the superhighway So if this is a newborn's intestine and you're taking, you're budding off the mom's immunoglobulin and you're going to send it to this cell membrane to give it to the newborn's blood, that process is called transcytosis. You know exactly how it happens. We talked about the vesicle trafficking. What do those vessels do? They don't sit around and wildly bump into each other. They attach to the microtubules via motor protein, and they hitch a ride, split ATP, and they go, and they just motor. They make that sound too. They just zip from one end of the epithelium to the other, infuse with the plasma membrane, and then deliver the payload, which is those mom's antibodies. Okay, now, meanwhile, what are all these little blue things? Notice the little blue things. These are intermediate filaments. You can think of them as cables that prevent this thing from being pulled apart. So, does the intestine get lots of activity? Absolutely. You're pushing all kinds of food and fluids. There's lots of activity. This thing would burst if it were not for these structural cables that are known as intermediate filaments. And they're actually anchored into places at the cell membrane and at the base of the cell by, these are desmosomes and these are hemidesmosomes, so they're membrane anchors. So this is really structurally very strong. You also find those same types of anchors in another tissue that gets lots of abuse, and that's your skin. Okay. People that have mutations in their intermediate filaments get all kinds of nasty blisters in their skin because it breaks really easily. Intermediate filaments are there for structural integrity. Okay, they're not for motor proteins, and there's a reason for that. Okay. All right, so we want to compare the three different fibers very quickly. You already know that the smallest one are these are microfilaments. They're composed of a single molecule, okay, that's stacked many times. Okay, the single molecule is called G-actin. That G stands for globular actin. And it has sort of a four-leaf clover look to it, and it's got a little pocket, which is a nucleotide binding domain. And the nucleotide that binds in that little area is ATP. It can actually bind ADP, but the preferred, the higher ATP is at higher concentration, so it tends to be ATP that fits up into the nucleotide binding domain. And so this is one actin, and that's right down here, and you just start helically adding actin, G-actins together, and you produce what? F-actin, filamentous actin. So you have F-actin, G-actin. It is sort of the basic reason this thing is so small. This is the smallest diameter. It's right there, 7 nanometers in diameter. Why is it so small? Because it's a single string of pearls. So that's the way to think of this structure. It's a string of pearls on a necklace going one direction. And it has a minus end and a plus end. Okay. The minus end is defined by which way the ATP binding domain faces. So all of these actins, their ATP binding domain is facing this direction, towards the minus end. The top up here is facing towards the plus end. Okay. And since we can't see that, this is too small, there's a way we can actually tell which direction the filament is oriented. Okay. We have a way of doing that. Usually it's towards, the plus end is towards the plasma membrane. That would be a good general rule. Okay, so the nucleotide is ATP. There's only one of them, and it's a string of G-actins strung together into a filament. So that's the structure of microfilaments. Okay, let's go up in diameter. And so we have intermediate filaments, okay? They're a little bit bigger, 8 to 12 nanometers. Just remember that they're intermediate in size. Why are they bigger in diameter? It's because the filament is a twisted rope. It's actually 8 fibers twisted into a rope-like structure. Okay, so you don't need to know the details of this polymerization scheme, but actually the fundamental polymer is a tetramer, and that forms protofilaments, 8 of them, that are then twisted into a cable. That's kind of neat because that's what you're using to keep your cells stuck together. It keeps them from being pulled apart. So you want a nice cable. Okay? There's no lumen in this cable, so it's like rope strands being twisted. So it's really strong. Okay? There's no nucleotides that bind to intermediate filaments. That's a big difference. There's no polarity. There's no minus, there's no plus. So, very different. Now, we got to get to, why is this the biggest one? That's 24 nanometers. Microtubules, despite the name micro, are the biggest cytoplasmic fiber in the cytoplasm. Okay, so here we go. This is the fundamental unit of a microtubule. It is a heterodimer. It is alpha tubulin and beta tubulin. These are related in structure. They both bind GTP, and it must be GTP. And so this is a heterodimer. Alpha and beta tubulin are synthesized by separate genes, and as soon as they are made, they come together, they stick. They have really high affinity to each other. You'll never split these two up. Okay? And so you have a binding domain here for GTP and one here for beta tubulin. So the way you make a microtubule is you take 13 different protofilaments and you form them into a hollow tube. And so that's why it's so big, is that's got a hollow diameter, a little hollow tube. And so it's very stiff. It's not flexible like microfilaments or intermediate filaments. So it's stiff. And that's good because you want if you're walking along a freeway or you're driving along a freeway, you want that to be nice and stiff and not to buckle, right? So that makes sense. This is going to be a molecular highway along which motor proteins are going to move. Okay? So you string them up. These are called protofilaments. You count them. There's 13 of them together. Turn them on the end. You can count those, right? Okay, so very different in structure. Different nucleotides. So now let's repeat what we know about the structure. We know that the difference is in diameter, and you know why those are different now. Okay. What about nucleotides? Microfilaments, ATP. Microtubules, GTP, right? Intermediate filaments, nothing. Okay. That's a good generalization. Okay, now let's talk about stability. The stability actually jumps around a bit. Okay, the least stable of all of these different types has nothing to do with their diameter. Okay, what it has to do is whether they bind a nucleotide. And the one that binds a nucleotide and is the least stable are microtubules. They grow very fast and they fall apart. And this little figure, let's turn the lights on just a bit, shows that in action. So you look, these are all microtubules. You look at number 3, and then you go over 125 seconds later, look at that. It's shot across there. Okay? So microtubules are very dynamic. They grow very fast, and they fall apart very fast. Okay? Let's find one that's fallen apart. So one is up here. The next frame, it's actually decreased a little bit, so it's gone down. And then look, it's then turns around and it's shot back up. So it's very dynamic. We call this dynamic instability. The only type of fiber that can exhibit dynamic instability is ones that bind nucleotides. And the most dynamic with the shortest half-life are microtubules, because that aids in their function. Okay. So these are the least stable, the most dynamic. And if we were to follow that, let's not go there yet. So the next, the one that's very unstable but not quite this stable, is more stable than microtubules, but it's less stable than intermediate filaments. That would be microfilaments. Why are they unstable? Because they have nucleotide bound to their gene. Okay? Nucleotide exchange causes dynamic instability. So the least stable microtubules, then you jump up here to microfilaments. That's the next least stable, less stable. Okay, and then the most stable is exactly what you would expect. If you're going to have a filament that produces structural integrity, you don't want it to fall apart. You don't want it growing. You don't want it shrinking. You want it to stay there. It will stretch, but it won't break. It's like having a great defense. It bends, but it doesn't break. But it has no nucleotides, so it's the most stable phase. Okay? So what determines dynamic instability? Binding of nucleotides. And I'll show you a good example of how that actually works. Okay. And before we take a break, what's going to happen at the break, Bailey's going to put out your tests, so give her a chance to stack those out here. Send one person down to come pick up your tests. But before we do this, the other thing is, and this is going to be the really core that you need to know for the final exam, because it compares all of the filament types. The last thing is the type of proteins that are associated with those microtubules, or with those filaments, okay? And this is actin, okay? Take a moment to commit that to memory. So there's lots of binding partners for microfilaments. The reason microfilaments have so many functions is because of all these diverse binding proteins. And what we call these are MFAPs, microfilament associated proteins. Microfilament associated proteins. There's lots of them. So there's not very many isoforms of actin, but there's a heck of a lot of binding partners. So they can do lots of different things. Everything from cytokinesis to skeletal muscle contraction to dragging vesicles across the cell. Diverse function because of the binding partners. Okay? That's also, and that's just an example of the different binding partners. We'll be talking about a few of those. Okay? So depending on what kind of binding partner, like if you're going to create a Bart Simpson hair, so this is a microvillus, you choose a cross-linker that's really short and stubby. But if you want to cross-link a skeletal muscle contractile fiber, you're going to have lots of space to put all the myosin motor proteins in between them. So you're going to have to use a different cross-linker. So the binding protein determines the function of the microfilament. That's all that's there for. This is an example of a binding partner for microtubules. So microtubules have lots of binding partners. Not quite as many as microfilaments, but there's a lot of them. These are all the different motor proteins that bind to microtubules. So there's lots of binding partners for both microfilaments and microtubules. Okay. The one, let me just, before we go on there, or before we finish for the break, what about intermediate filaments? Very, very few binding partners. The diversity of function for microfilaments has to do with all the isoforms, which are very diverse. So microfilament functional diversity comes from the number of different ropes that can be made. Okay? And it's almost different in every different type of tissue. The intestinal microfilaments are very different from brain microfilaments. And so we actually use that characteristic to type tumors. So if you get a tumor cell that's in the brain, it may not have started there. You can actually, if you take a biopsy and look at the type of intermediate filaments it contains, and it turns out they're intestinal intermediate filaments, then you know it probably metastasized from the gut. So it might have been a colonic tumor. Okay, so we can actually type things by the type of intermediate filament that they have. But these two guys, the microfilaments and microtubules, there's not very many isoforms. It's the binding partners. Okay, let's take a break. Give Bailey a few minutes to get these spread out down here. Yeah, that kind of went through that fast.
So the insulin receptor is, like the alpha subunit, is regulatory?
Yes.
Okay.
And it is the part that binds insulin.
Right.
Right. And without insulin, it's actually an allosteric regulator. Negative allosteric regulator.
Okay. Okay. And also about how long on Tuesday next week, or next time we put some presenting practice? about?
Oh, no, we won't be presenting next Tuesday. We'll have one more lecture. And so we start presenting a week from this coming Thursday.
Okay.
So one lecture left. We're going to finish up a little bit of the cytoskeleton, then we'll do cell cycle. That'll be it.
Okay, and
Thursday will be cell cures 1.
okay, and how long? Like
It's about 8 minutes.
8 minutes for each group.
Yeah.
Okay.
Each little set.
Right.
Right.
I was just curious because my group had like kind of put together everything very
So that's pretty much what you said. You know, it might be a minute more or less, but it's about 8 minutes, and then that leaves, I think, 1 or 2 minutes for questions. But people are actually encouraged to ask questions throughout. But I'll send some details about this. I just haven't gotten to it yet. But,
Okay.
but that's pretty much what it's going to be.
Okay, good deal. Thanks.
And so if you have, somebody's going to have to present it 8 times. Because you have 8 groups that are presenting and 8 groups that are viewing. And so 8 minutes are up, then they move to the next station, and then the next person in your group. You can't have more than, I really don't want 2 people presenting at the same time. Like they do the first half of the poster. Each person should present the entire poster. Because you could have like, okay, person number 1 just does the introduction. Person number 2 does the objectives and the hypothesis. And person number 3 does the rest. I don't want that. For every time it gets presented, the person must do the entire poster.
Okay, so.
And that's what takes
that
8
changes-
minutes.
That changes everything.
So what, you were going to break it up and have people do different things?
Yeah,
You can't do that.
okay.
Because what happens is people learn only one part of their own poster. That's why. I want everybody to learn every bit of their own poster. Now, what can happen is a person is presenting and they start to flub up, they can get help from people. And so you should stay engaged. And also, if they get questions they can't answer, that person should try, and if they can't, somebody can step in and help. So you get help, but everybody has to know the whole poster.
Okay. Then
And if you have some people, how many people, group members?
we still have 7. I
So
mean, like, I think.
somebody's going to have to do it twice, and you get to pick that person.
Okay, well,
Does that help?
yeah, I'll
Yeah, we had one group that had 2 people in the group. They had to present 4 times each. Yes. Pardon me? Oh, really?
If it was like 11:30 or something, I could like go pack up my car.
Where are you from?
Dallas area.
Oh, what part of
I don't think
I'm from
waste
actually
time all the
Garland,
way.
so like North Carolina too, so it's not too far. I guess
What?
for some reason.
I must have just put them off.
Yeah.
Because I know I have. Are you?
Yes.
They're going to be outside my door.
Oh, well, we have an appointment to meet you, our group.
So you just come up and get them. Yeah.
Oh, I had a question. I'd emailed you about meeting too, but I had checked my email. I hadn't seen a response. Is there any way it could be before office hours today?
Well, I'm meeting with the group right after. Can you meet right after?
Yeah, I can meet right after you meet with them. Okay, that would be awesome. Thank you so much.
Okay.
We have a question. Did grad students have to do the grading of the grade?
I don't think you guys were sent them. No.
We all got it,
You did?
but it's not on our, it's not showing, we got the email, but it's not showing up on Blackberry.
Let me.
We have a question on that.
Okay.
We're kind of doing unemployed, but in order to find it, we're pretty much seeing like we know that there's this complex that is the exact location on the chromosome, and so for our methods, what we want to do. Remember, I had mentioned to you about gathering one cell specifically, and then phosphorylating a specific location on the chromosome.
Yes.
I mean, putting fluorescent light on it to basically see what occurs. So if we were to
Like a laser.
yeah, like kind of like the antibody, just kind of like
So the antibody probably has a fluorochrome.
yes,
And then you light
put
it up by
on the
hitting the
exact
laser
location.
light.
Yeah. on the exact location of the chromosome. And then if we see pretty much the synapses occurring, and then we can identify the locations that interacted within that complex that we're looking at, and then see if it affected to become aneuploidy or not.
Sounds good to me.
We can do that. Okay. Do we have to find a solution, or just pretty much like
Well, you could just say,
Say
we're going
that's
to, here are some possibilities
that we
of what
try
might
to?
happen.
Okay.
That's where that little discussion comes in. 'Cause people will say, well, what if this happened? And you go, well, we thought of
There
that.
could be
This-
multiple like things
Outcomes.
that would affect it. Okay.
Right. Okay, we need to sort of get started again. Okay. So now one of the learning goals is to distinguish between how things grow in a test tube versus how they work inside of the cell. They're very different. And what we're going to start at looking at microtubules is our sort of poster child for that. In a test tube, well, and this is actually a picture of actin filaments, but it really doesn't matter. Both microtubules and actin filaments do the same thing. It takes forever in a test tube just full of microtubule, alpha beta tubulin, or actin for them to get started. So that's this nucleation phase is the longest thing. Okay, and then once they get started, it starts to go pretty fast, right? And then it reaches some steady state that's completely determined by the concentration of monomers. Okay, whether it's the concentration of alpha beta tubulin or the concentration of actin, G-actin, that determines the length of the filament. This is entirely not how it works in the cell. So this, you can just kind of scratch this whole thing. Actually, what happens is the rate of nucleation is completely controlled by filament-associated proteins. So there's something that helps nucleate this, there's something that helps elongate it, and there's something that actually stabilizes the filter of the filaments and length. So it's all about the type of binding proteins. Now, which of the filament types could you say that's not going to happen? It's not going to be controlled by the associated proteins that bind to it. Intermediate filaments, because they don't have very many binding partners. So it must be we're not even going to talk about that, because people don't really know a lot about what controls intermediate filament, polymerization, nucleation, and the fiber length. So we're going to sort of leave that out. But for these two, microfilaments and for microtubules, it's completely controlled by their binding partner. So that's a good thing to know. All right. So let's talk about how you construct this, and the way you don't construct it is to take each of these protofilaments and kind of stick them together. They actually grow in sort of a circle, okay, as it grows up. This is called a singlet microtubule. It has 13, if you turn it on its end, you can count these numbers, and there's 13 of these protofilaments that are stuck side to side, okay? All right, and so what controls the polymerization? The polymerization, or what actually is regulating the stability of this microtubule? You know that these are the least stable, and the thing that controls stability is the fact that it binds a nucleotide, and the nucleotide that's shown is only the beta GTP, the GTP bound to the beta, because the one in the alpha is never touched, it's never hydrolyzed. But this is actually a GTPase. Okay, so the beta subunit can actually hydrolyze its GTP. So what happens is you can't add an alpha-beta tubulin dimer unless it's bound to GTP in the beta subunit. Okay, so as long as you're doing that, so you keep adding these, you get a string of GTP beta tubulins, right? Now what happens is after a certain amount of time, the GTPase kicks in and it starts to, this is the first one that was started, so that will click in and that will turn to GDP. And then after a while, that will turn to GDP, and then that will turn to GDP. And when that GDP reaches the end of the protofilament, that's when you so-called lose the GTP cap. As long as you have this capped off with the GTP beta tubulin, this thing will not fall apart. But as soon as that reaches the edge, it goes kablooey and undergoes what's called catastrophe. And it goes like that. That film that I showed the very first day, you saw that thing unpeel, that was called catastrophe. And what that meant was the rate of GTP hydrolysis has exceeded the rate of polymerization, adding new GTP tubulins on top of it. So what you get is this. And so if this falls apart, the alpha beta still remains stuck together, then they can be reassembled into a new filament as long as you exchange the GDP falls off, the GDP goes on. So this thing is now fully loaded, and that can go back and be added to a new protofilament. Okay, so the key is that's called the GTP cap, because everything that's facing outwards is a beta tubulin that's bound to GTP. And what's happening is this is continuing to try to get, these beta tubulin, which is the dark green, is splitting its GTP into GDP. So these are all GDPs, right? And you can see it's gotten up here, but you still have a cap of GTPs, and so this is still stable. But as soon as all of these guys split their GTP, that is, you're not adding any new alpha beta tubulin dimers, then this thing will undergo, it'll just peel apart. Okay? And actually the reason it does is because when it turns to GDP, it starts to bend. And that pulls the microtubule apart. And so then it does this, it just folds out like that. Now, it's not going to completely go all the way back to zero, it's going to fall back. And then based on the concentration and the type of proteins that are around, this thing will reform. Okay? So the key about growing and shrinking is the rate of growing and shrinking is always faster at the plus end. That's what that plus tells you. Think of it as being positive. Of course, it'll grow faster, but then it'll also collapse faster. Okay, so it's the most dynamic end. The minus end is sort of where it began its story, so you really don't want to fool with that end too much. So the minus end is always pointed close to where it was born, or in terms of that, it's nucleated. Okay? And that's always near the nucleus. And there's a reason for that. Okay? So which of this end is plus? It's always that end. Okay? And actually, the tubulin that's facing the minus end is always alpha tubulin. And if that, see, there's an alpha beta tubulin dimer, it's the beta that's always facing towards the plus end. And so it's always alpha beta, alpha beta, alpha beta. Okay? As I describe up here. So that's shown right here. Minus, and it's alpha beta, all the way to the plus end, where you have beta facing the most plus end. And that's usually projecting towards the plasma membrane. So the minus end is usually near the nucleus, the plus end is towards the plasma membrane, but not always. Okay. So a lot of anti-tumor drugs are obviously things that mess with microtubule polymerization in some way or another. And one of the most effective, particularly in breast cancer and a few other solid tumors, is this chemical called Taxol. It's a really obviously complicated molecule, and it comes from the Pacific Northwestern yew tree, Y-E-W. And actually, in the old days, you made that chemical from the bark, but that killed a lot of trees, right? And so now they can actually take the leaves of the yew tree and convert it into Taxol without having to take the bark, which kills the tree. So for a while, they were deforesting all of the yew trees in the Pacific Northwest. We don't have to do that anymore. Okay. So we have a renewable source. And what does Taxol do? So here you are, a normal cell and a great microtubule kind of sticking everywhere, you know. You throw in Taxol, what it does is it stimulates polymerization. All of them. All the microtubules are thrown into polymer state. Now, the key, why does that block mitosis? The spindle has to be very dynamic. It has to reach out, grab the chromosomes, and it has to depolymerize to help bring it back to each side. If you stiffen that place, there's no way they're going to attach to the chromosomes, and no way they can retract and segregate the chromosomes. So this is going to kill the tumor cell. Unless they express MDR1 and blah, blah, blah. Lots of different ways for this to be defeated, but it works very well. It's also very toxic. This is a brute force non-directed chemotherapy that's meant to just kill everything that's dividing. Okay, but it's very effective. Okay, so, and then there are other drugs, such as colchicine. Let's see if I can talk about those. Yeah, one of the classic drugs, this actually came first before Taxol. Colchicine actually binds to the tubulin dimers before it goes on to the fiber, and once it goes on to the microtubule, you can't form any more. So this actually blocks mitosis because you can't build the spindle fibers. So Taxol makes them all polymerize. Colchicine prevents the polymerization. So either way, you're going to kill the spindle fiber, and that's the point of a lot of chemotherapy, is to block the dynamic activity of spindle fibers. Okay. Now, I mentioned that proteins in the cell actually nucleate the start or the growth of microtubules, and that's what happens. So that place where that happens is called the Microtubule Organizing Center, MTOC. And it's down here, okay, MTOC. So the generic term is Microtubule Organizing Center, but this in particular is located next to the nucleus. It's called the centrosome, and it has sort of a little halo. There's no membrane here, but there's a halo of centrosome matrix that recruits certain proteins. And the main key ingredient is this protein right here, the gamma-tubulin ring complex. It's called the gamma-tubulin ring complex, gamma-turk. It's like a little circle, and it's made out of gamma-tubulin. So there's not many isoforms of tubule, but there's a few. And the gamma-tubule is what nucleates tubule. Okay? And it's located, associated with the centrioles. Okay? So everybody has a pair of centrioles near the nucleus, and they're all sort of at right angles, as you can see. But the centrioles themselves do not nucleate the microtubules. It's the material that they recruit, which is the beta-gamma ring complex. And it is actually shaped like a ring. So here's this sort of weird structure. This is the beta-gamma thing, but it's kind of like a circle. And so these protofilaments start to grow out, and then they associate side to side, and pretty much this thing spirals outwards and actually zips up the side and becomes a full microtubule. But it all starts at the negative end of each one of these protofilaments. That, I can guarantee you, is alpha tubular. The dark is beta, alpha, beta, alpha, beta, alpha. It must end in beta tubular. It must start with alpha tubular. Okay? So, that's the centrosome is a type of microtubule organizing center. There are some microtubule organizing centers, as you saw in the intestine, enterocytes, that are located elsewhere. And in particular, cells that have cilia and then are flat flagella, like sperm, and then the fallopian tube that has lots of cilia, those are projections of microtubules, right? And they originate just underneath the cell surface. Let's see if I have a picture of one. Yeah. Okay, that's just a picture of the centrosome. Okay. Looks like a spiky sea urchin. Okay. And one of the things that's characteristic about microtubule organizing centers is that centriole, even though that's not what's nucleating the microtubules, they are themselves composed of very special microtubules. They're actually triplets. We call them triplet microtubules. And they're actually rings of, there's three of these. The A ring is the only one that has 13 protofilaments. The B ring and the C ring have 10. But the key thing is they have triplet microtubules that make up the centriole. That's how you know it's a microtubule organizing center. Is it the centriole that's nucleating this? No. It's recruiting the beta-gambula, the beta-turk ring complex to it, and that nucleates this. But if you go and you do a section through this electron microscope, you can look at the diameter, and you can say, that's a centriole. Okay. Now, at the base of the plasma membrane, that's a different type of MTOC called a basal body. Those are microtubule organizing centers that give rise to flagella and cilia, and they're located right under the cell membrane. Do they look like this? Yes. They have triplet microtubules. So all MTOCs have triplet microtubules in most cells, but there are a few differences. I don't think plants have centrioles. I don't know. I may be wrong about that. Yeah. Okay, so the key thing is microtubule organizing center recruits the gamma Turk ring complex, and that's what nucleates the microtubule growth. Okay, I'm actually going to stop early there and pass out class evaluations. So I really would appreciate if you would wait and get those and fill those out, because a lot of my colleagues don't read them. I do, okay? So please take some time to fill those out. And I need some helpers. Maybe you could come down and help. Get a couple of graduate students, maybe a couple of you guys to come pass these out. But please do wait around to fill those out, and I'm going to try to get out of here.
So should I wait to return the pad, or should one of them go with her?
Oh, you can help. You can wait. Help. Just give it to somebody. Oh, you can turn it. Yeah, that's fine. You're not going to look at them. And they're not evaluating you, they're evaluating me. Just be sure to throw the bad ones out. I'm kidding. Yes.
Do you know the course number and section?
The 30 undergraduate? Yes, 33. Section 1.
Thank you.
Yeah. And graduate students 5302, section 1.
separate piece coming from the where did this one? So all this one goes to one side and all the other yeah, it doesn't matter.
I gotta be my point, but I pretty much point good. Are you sure I did that part?